Good morning, everyone. Thank you so much for coming to celebrate with us Yom Yerushalayim, in which we commemorate the reunification of Jerusalem after the Six-Day War in 1967. My name is Ifat Biran. I'm the Victorian coordinator for the Australian Friends of Hebrew University, which just happens to be in Jerusalem. In fact, the Hebrew University was founded in 1918 by the likes of Albert Einstein, Chaim Weizmann, Sigmund Freud, and Martin Buber, and is also the only university to boast 8 Nobel Prize winners. Hebrew U is world-renowned for its excellence in a variety of areas, including molecular neuroscience, which we'll hear about by Professor Hermona Soreq. And the Australian Friends of Hebrew University is proud to represent Israel's #1 university and the highest ranked Israeli uni in the world rankings. The prime role of the Australian Friends of Hebrew U is to raise funds for research and projects at the university, but it also promotes short and long-term programs for gap year and university students, for which I've put some flyers for you to have a look at, as well as research collaborations between the Hebrew University and Australian universities. If you wish to learn more about the work that we do or any of our programs, feel free to reach out afterwards. But it's now my pleasure to introduce to you our remarkable guest speaker, Professor Hermona Soreq. Professor Soreq holds a University Schlesinger Chair in Molecular Neuroscience and is a founding member of the Edmond and Lily Safra Center for Brain Science. She is the elected president for the International Organization of Cholinergic Mechanisms and former dean of the Faculty of Science at Hebrew University. During her outstanding career, Professor Soreq has trained numerous scientists in academia, public and private sectors, and has been the author of hundreds of publications, including many in high-ranking scientific journals. For her academic achievements, Professor Soreq has been given a long list of honors and awards. I won't go through all of them, but they do include the 2022 Israeli Prime Minister's Emet Prize in Neuroscience. Today, Professor Soreq will tell us about how female and male brains react under mental insult. And just to give you a brief overview, we all know that everyone is subject to stress and mental insults, but men and women react differently. Schizophrenic men are addicted to smoking. Schizophrenic women are not. Women with bipolar disease suffer more acute depression than men. And women also respond more drastically to many brain-affecting medications. Professor Soreq and her team discovered that these variations are a result of distinct profile of molecules called small RNA. As an example, women with Alzheimer's disease deteriorate cognitively faster than men due to a loss of particular group of small RNAs in their brain. With the recent technological advances and the FDA approval of RNA therapeutics, i.e. COVID vaccines, amongst others, there is now hope that RNA-based drugs will change the standard of care of many mental and other diseases. Actually, Professor Soreq's pioneering research on the neurotransmitter acetylcholine and its RNA regulators has already led to the development of drugs at the Hebrew University for the treatment of Alzheimer's disease. Please join me in giving a warm welcome to Professor Soreq and enjoy her fascinating seminar.
Thank you, Ifat. That was overwhelming. I actually have a grandson whose friend in school said, "Come on, nobody has a grandmother in Wikipedia." So he could find about me just like Ifat. So thank you for inviting me to be here, and I'd like to talk about the reaction to mental insults from brain to body. And let's start with a Bible that says about feelings, hamu b'nei me'av. That means intestinal reactions to feelings. So we all know about it, but we don't really think about it that focusedly. So that's what we want to talk about today, and ask how does the brain initiate that? Which are the genes that control it? What's the outcome? And what does it mean to the difference between men and women in reactions to numerous diseases affecting the nervous system, and all that in light with the Jerusalem Day. So I looked for pictures for you, and of course everyone knows this street in Jerusalem, and it gets to the question that we would like to start with. What are mental insults? When and why anxiety and inflammation empower them? So if we look in the literature, we find that susceptibility to inherited phobias may be quite prevalent, and some of those phobias may be acquired. And those relate to nerve cells that react to chemicals. So these are what we call neurotransmitters, chemicals that are brought. And I was once, for example, asked to come to television and talk about the fear of flight of Yoram Gaon. So now you know he's afraid of flying. I said, that's not my field of expertise. I can't talk about fear of flying. But when we think about mental insults, people come to mind like Virginia Woolf, of course, who was a very, very sick woman. We remember her wonderful creative literature, but she actually committed suicide by putting stones in her pockets and going into the river. So that's one example, but there are many. If you think about people who are subject to anxiety, a lot of the very well-known heroes of the previous generation are known to be anxious. So Woody Allen made it really known to everyone, but most of the others didn't. And you think about people like Mark Twain, who would be anxious? I don't know. So it's much more common than we think. About 10% of humanity, we are saying, and these are normal days. God knows what happens after COVID. So just to change the atmosphere, this is one of the gardens in our campus of the Hebrew University. I'm often asked, how did you come to the Hebrew University? And I came when I was 15. I was studying in high school in Rishon LeZion, and my chemistry teacher came and said, there is a summer program at the Hebrew University for interested youth, and you'll be a biochemist. He really did say that. He's very proud of that too. And he sent me for 3 weeks to the Life Sciences Institute, where I'm working today. And I loved it. I loved every minute of it. We lived in a little place in Kerem. That's our village today, so that's where our house is. And after 3 weeks, I came home to my parents in Rishon LeZion, and I said, don't we have any relatives in Jerusalem? The lab members would like me to stay, but we don't have where to sleep. So sure, everyone has a relative in Jerusalem, so they found this aunt, and I stayed there for the rest of the summer, and that was it. I came home, I was hooked. And here I am, still at the Hebrew University. So let's get back to mental insults. So people come down with post-traumatic stress disorder. It's quite common in a country where wars happen every second year or so. And the drugs that are admitted affect not only the brain, but also the body. So I have friends in Berkeley, whenever they are to teach, they take a beta blocker to calm down the pulse. That's a mental impact, but it works through the heart, okay? And this is Karen, who became an expert in convincing people in the qualities of medical equipment after she completed her PhD at the lab. Now, if I told you about acetylcholine, so I'd like to share with you some Why am I interested in that? So this guy has been Otto Lowy. He was a Jewish professor in Marburg, in a South Germany university, that didn't give him a position because he was Jewish. So he moved to Graz and in Austria, and there he showed that the vagus nerve, which comes from the brain and into the brain, so there is vagus going down, vagus going up, activates the heart. And at that time, it was still thought that the nervous system is like an electrical system. So one nerve touches the other and sends a spark, and that's how our brain is activated. And he showed that if he could put one heart into high activity by an electrical wire, take the liquid of that heart and put that on another heart, that heart starts palpating. So it was a chemical, and he could identify the chemical, and the chemical is called acetylcholine. You see the structure right now. And he won a Nobel Prize for that. So that was 1906 when he won the prize. I learned all that in Marburg University that celebrated 100 years to this discovery, to amend that they refused the job. Okay. So he was working in Graz, and then in 1938, Germany took over Austria. And suddenly he became a Jew working in a government system, which was not allowed. So he was thrown into a camp, and he kept sending postcards. There was a historian in the conference who showed those postcards. He was sending his students instructions which experiments to do next week. So, and then the speaker said in Marburg, the German government was in a financial hardship, right, at 38. They were getting ready to conquer the world. And they reached an agreement. They took the Nobel Prize money, and they gave him a one-way ticket to New York, where he completed his career and died at a long age. So he also found chemicals or plant extracts that could activate the heart, and these are parts of the treatments we give today to Alzheimer's disease patients, because one of the first systems that is damaged there is the network of acetylcholine. Okay, so just brought you the Nobel Prize. In that time, they were hand drawing the announcement. And this is how he ran away. Okay, so let's now get back from this story to where does anxiety come from? It actually has three interactive sources: our genome, the environment, and personal experience. And here's a citation of Schopenhauer, who said, even earlier than Otto Lowy, that the niceties make the difference, but fate gives us the end, and we play the cards. So each of us carries a DNA that is slightly different from anyone else, except for identical twins. And some of those DNA marks may be not that good for us. What we can do is try to make the best out of that. So the questions we're going to ask now is, are there susceptibility genes that anxiety is activated by? So you think about DNA, two strands with some areas being altered, so different in one person compared to the other. Could those relate to the cholinergic network? That was the question I started asking when I came back from New York, where I was a postdoctoral fellow. And we looked at the cholinesterase genes. There are two genes, and we cloned both of them. And the way mutations in those genes affect anxiety reactions. But it's not only anxiety reactions. If we think acetylcholine is the culprit, then insecticides are targeted against the same protein, acetylcholinesterase, And we share about 50% of the sequence with insects, which means that insecticides are not very healthy. If you see a spraying airplane, don't stand below that. It's not good. And the picture of this brain shows you a brain map of people who live next to Beersheba in areas that are sprayed consistently because they are full of orchards. So they're not exposed personally, but they live in an exposed environment. And what you see here is that their frontal lobe is hyperactivated, the red mark, which means they are anxious. Worse yet, the nuclei in the brain, the down nuclei, are suppressed, and they suffer learning and memory impairments. So exposure to insecticides is bad news, and it would be even worse news to people who carry mutations in the genes. Not only in regions that encode proteins, which was a very big puzzle for us initially, but today we know that these are the regions that interact with small RNA regulators that we are getting to talk about in a minute. So we have a big friend at our brain center, Professor Eric Kandel from New York, who also left Austria as a child and won the Nobel Prize for his findings. And he keeps saying that psychiatry will never become a serious field until they understand genetics and use those tools. Okay. So what do? We look at these small RNA regulators that work like traffic lights. So let's think about gene expression as a pyramid. There's DNA on top. You don't want to meddle with that because God knows what will that do to the next generation. There are proteins in the bottom. They do all the work for us. And in the middle you have RNA. So these are copies of parts of our genome, and they can either produce a protein or control the production of protein. And we study those RNAs that control the production of proteins. You see the little red thing here is wrapped around a green thing. The green thing is the RNA producing a protein, and the red thing is the regulator. Small RNAs that work like traffic lights. And this is Shachar Barbash, whose PhD showed that 1/3 of the small RNAs in primates, to which we belong, are different from those in mice. So you can't really say, I'll study mice and I'll learn everything about human beings. So acetylcholine is an important parameter in that control by small RNAs. And let's get to the anxiety. How do you check reactions to anxiety? So you can find papers in the literature. One paper says about this guy who experienced a massive earthquake and developed a heart attack and dropped dead. Another experienced a tsunami, and so on. And then the discussion part in those papers often says you cannot study anxiety in big populations because there are no populations that are consistently exposed to anxious conditions. I disagree, humbly. I think that I come from a country that is consistently exposed to stressful conditions. So we decided to look at that level in healthy individuals. And we studied 18,000 volunteers who come annually to a routine medical checkup paid for by their employers. And we added a questionnaire: to what extent do you feel the impact of terror attacks on your daily life, from 1 to 5? That kind of questions. And then we thought, what can we check in those people which would reflect anxiety? And this is the simplest test possible. Put your thumb on the other hand wrist and count the number of times your heart beats in 1 minute. So that's what we call pulse, right? It's a very simple no-cost test, which is why very few research projects deal with it, because it's too simple. But if you do that to 18,000 individuals, you can find out not only what was their pulse, but since they come every year, you can find if there was a change over those 4 years where we ran the study. And we, in that case, were a postdoctoral fellow, Dr. Shani Shenar, and 2 PhD students, Nadav Yayon, who's now a postdoctoral fellow in Cambridge, UK, and Nir Weisskopf, who runs a small startup company. And this is what we've done. We measured the cholinesterase activities and we measured pulse. And then we applied machine learning to ask how many of these individuals saw an increase in their pulse over the years of the study. Now, a consistent increase in one's pulse is bad news, because it means that your heart doesn't work as efficiently as it should. So anyone who sees a consistent increase in their pulse should address the family practitioner, because this is an increased risk of stroke and heart attack and death of all causes, the papers say. So when we looked at that, about 700 of those individuals had such an increase in their pulse. Then we addressed their questionnaires, and what we found is that those people also filled those questionnaires of fearing a terror attack 5 out of 5. So if you're anxious, your heart rate is changing and not for the better. So we sent that paper to the Proceedings of the National Academy of Sciences, very prestigious journal, and Shani says, so what happens next? I say, okay, so every day you come to work, you get on the website, you find no change, and one day there will be a change and there will be the reviewers' responses, and they will tell you what you need to do to improve on this study. And this is what you'll do the next half a year, except I was wrong. At that day when we had the answer, it said that the authors should be uploaded, and the paper was accepted with no corrections, which is really unique. Okay, so volunteers who fear terror attacks are more vulnerable to pulse changes and also inflammation. The inflammation biomarkers in the blood also went up. So this is a cardiac riskful condition, and last month there was a paper that proved that same phenomenon in engineered mice with very complicated technology, so now we know it must be true. Okay. And that is also what happens under a COVID infection. So if we look at that, inflammation markers go up, the cholinergic system is impaired, people become more anxious. You can't find a child psychologist in Israel today because they're all over-occupied. And we had only 2 months of closure, so I guess you guys have even harder conditions. So yeah, I found a gene that is modified that is called dancer, so I couldn't help myself. This is Nimrod Madrer. He's a PhD student in the lab. And this is Hanan Meidan, who's a clinician that treated COVID patients. So that was that study that showed the impact on inflammation. Okay, so let's agree, I think, that anxiety induces inflammation and inflammation induces anxiety, so we have a vicious circle here. Not only the head, but also the body. And this is Geula Hanin, is also a young scientist today at Cambridge, and she looked at the association of all of these systems together. So, what are small RNAs? The leading company of small RNAs are microRNAs that were discovered in the beginning of the century and got a Nobel Prize to these 2 Californian scientists. And we looked for those small RNAs that control the cholinergic pathway. And we found one, we were very excited about it. Let's see if the movie works. No, this movie didn't work, but you saw it before. So this is the microRNAs, the red thing, and the RNA for acetylcholinesterase is the green thing. So we said, let's find out what happens if you block the microRNA. We took a mouse and we cut off this region which reacts to microRNAs. And what happened to the mouse is this. I don't know how many of you ever saw a mouse behaving, but they sit and they scratch and they look for food. These mice run, they all run all day. You'd say that's very healthy, they do sports. If you put them into a maze where there's apple juice at the other end, the healthy mouse runs to get the apple juice really quickly, whereas the engineered mouse, I hope you can see that, he goes back and forth, he forgot its glasses, he doesn't know where it left the keys. I should be very careful with this comparison because one day a woman in the audience said, just like me. Anyway, these mice are anxious and stupid. And all that is because the microRNA system cannot function. And they also show inflammation. So that, of course, raised the question, can that happen in human beings as well? So Shani again collaborated with clinicians in the Soreq Medical Center in Tel Aviv, and they operate on people with Crohn's disease who suffer severe inflammation in the intestine. They remove those regions of the intestine, and they remove them until they get to a non-inflamed domain. So we looked at the levels of the microRNA in inflamed and non-inflamed human tissues, and it's like tenfold higher. So inflammation and anxiety both elevate those microRNA controllers, and I was really excited about microRNAs. I kept saying, you know, they're so clever. Small genes, all you need is to activate a promoter and you get a real useful tool. Someone should have invented them if they didn't exist. So what happens in neurodegenerative disease? MicroRNAs go down in the brain, and especially microRNA 132, the one that I just talked about. So the levels there are inverse, and the brain cannot really function without microRNAs. So this is the beginning of our study on Alzheimer's disease. We measured the enzyme that is targeted by that microRNA in the blood of patients, and we found that the levels go up even before they become fully blown Alzheimer patients. So that's one microRNA. Are there other examples? So, and this is Mor Hanan. She runs a small startup company in Rehovot now. Okay, so let's talk about another type of reaction of a microRNA. In 1998, the Japanese television system broadcasted one section of the Pokemon series, which had been very popular with children worldwide. Except that that section had a very rapid lightning issue. And that night, 800 kids were hospitalized with acute epilepsy in Japan. So that program was taken off. It can be found on the web, and it's really unpleasant to watch because of those lights. But for people who are susceptible to epileptic seizures, I mean, that is a real trigger. But that raised three questions in our mind. First, what's wrong with those 800 children? Why did they get sick when like 20 million kids watched it? How did the other ones get protected? And last but not least, it only took like 7 hours. To activate a gene, transcribe, send over, takes much more than that. So how come this was such a rapid response? And this was the question that Uriah Bekenstein asked with Niba Mishra who was a postdoc that came from India to the lab. And they put through an opposite theory. They said, what if there are microRNAs that are there to protect us? And if their levels go down, then we are not protected. What if there's something like that? That's a small RNA. It can happen quite rapidly, much more rapid than a regular gene. And they looked for small RNAs that might fit that perception, linked to the cholinergic system, and found a microRNA 211. They all have numbers but no names. And they engineered a mouse where the levels of that microRNA were high, except if we fed them with antibiotics, in which case they go down. And when the levels go down, the mice became epileptic. So, okay, we proved one part of that concept, and now you should ask me, so what comes out of that for Shabbat? I mean, is that useful? Which is a question we often ask for scientific articles. And the problem is that 20% of Alzheimer's patients are epileptic, and this is not even treated. So we looked in the brain tissues from Alzheimer's patients, and indeed that microRNA is much higher up in their brain, and that's probably the reason, which again opens up an opportunity for treatment. So Alzheimer's is the problem, and our studies show that there is a change in that microRNA. Which brings me to men-to-women reactions. So this is a study we ran between Geula Hanin that I mentioned and Sebastian Lobentanzer, who was a PhD student in Frankfurt University, with a friend of mine, Jochen Klein, and he kept coming to every conference in Germany I was invited to lecture in, and we started collaborating, and then one day I get a letter from Frankfurt University about your PhD student, Sebastian Lobentanzer. Okay, so I became his co-supervisor. And what we've done is to ask the questions, to what extent men and women reactions to mental disease are the same. And we did that through the cholinergic system, surprise, surprise, and with the hope to look for some interesting solutions. So we compared the reactions of men and women with or without Alzheimer's, a mental disease, so schizophrenia and bipolar disorder. Now, as Ifat told you in the introduction, we already know that schizophrenia is subjected to impact on the cholinergic system, because schizophrenic men are always addicted to smoking, and nicotine activates acetylcholine pathways. Women don't. Women, on the other hand, suffer very severe impact of bipolar disorder, much more than men. And there is no relevance to any of the medications in that. The psychiatric treatment involves trying one medication. If it doesn't work, they would switch to another. But very few studies looked for that initially before treatment. So here is what we have found about the cholinergic system. So this is a comparison of schizophrenia to bipolar in men and women. And here you can, if you wish, see the pathways that are affected, including response to organophosphates, which are those insecticides we started with. And you know what? There is a guy named Alexei Navalny that was poisoned 2 months ago with exactly the same kind of poison that is used as an insecticide. And I hear that he's very, very sick. So in schizophrenia, you see different pathways going different directions in men and women. In bipolar, it's the same pathways, but opposite directions. So there is a huge difference between men and women, and it relates to the cholinergic system. So what do we know about the brain and the cholinergic system? This lady was Rita Levi-Montalcini. She won the Nobel Prize for discovering a protein called nerve growth factor. And she created a company, I think in Sweden, that aimed to develop new treatments to the brain based on that. But if you look at the brain, okay, there are different systems that work via acetylcholine, from deep brain nuclei all the way to the cortex, and they are very different. And Rita Levi-Montalcini lived before the World War, so she wouldn't know that. And that company didn't succeed, but we are still interested in looking, what about the cholinergic genes in men and women with mental disease? And we started looking at that in cell cultures. Now, cell cultures are just one line of one cell that can be multiplied. And we know, for example, about Helen Lane, who suffered cancer, and her tumor served as the origin for cells that everyone uses in research. So that is a woman-driven cell line. Other than that, we never think about cells as coming from a man or a woman. It's like we treat them as if they're test tubes, empty. And that's wrong, because when we looked at neuroblastoma cell lines, so a neuronal tumor, from a child and a little girl, we could see very different patterns of microRNAs, those small genes. So we could see some that were specific to males, others that were specific to females, and others that were joint, and others that were opposite. Since then, whenever I use a cell line, I first ask, does it come from a man or from a woman? And when we treated these cells with acetylcholine, so that to drive the cells to improve their capacity, you see a change in all of the pink points show you women-specific microRNAs. So this is a woman-derived cell line. And with a man, you would see different ones. But microRNAs each controls different genes. So when we looked at those genes that they control, we find that many of them control schizophrenia, others control bipolar, some are dual, very different. Now, I have to tell you that I had a very difficult time publishing this paper. The editor of the journal, of course, sent it to an expert reviewer. And the expert reviewer said, this is magnificent. You find a new type of cholinergic neurons, that's great. You show us the networks, this is wonderful. Just forget the difference between men and women and I will accept it. Luckily, the editor wrote me, I actually think that this should become a resource for others to learn the difference. So that finally happened. We published the paper, and then I got an invitation to write a review about the differences between men and women in reaction to drugs. And apparently, most of the basic research studies are done in male mice because it's cheaper. You don't have to control for the estrous cycle. In female mice, you'd have to use 4-fold larger amount of animals, which is very expensive. Then it comes to clinical trials. Women tend to volunteer less to clinical trials than men. And eventually, it's sent for treatment, and only then people start following the differences. So just before the COVID, in January 2020, there was a conference of the Israeli Science Foundation together with the Chinese one. So we all flew to Shanghai as a delegation. And I see my friend from the Sheba Medical Center getting into his chair, taking a pill, and he's asleep for 8 hours and wakes up beautifully. I said, Shai, how do you do that? I never sleep throughout the flight. And he says, oh, no problem. I'm a doctor. I'll give you this drug. But remember, you have to take half a pill. Because 2 years after this drug was approved, it was found that women that took it the evening before were responsible for traffic accidents the morning after. So this is what happens in reality. Drugs are developed for males, tested in males, and released for use. Half of humanity is getting treated by drugs that were developed and tested for the other half. So what we see here is that the levels of a drug in the blood might be different, the levels it reaches in the brain would be different, side effects, bone fracture might be different, epileptic reactions, I mean, the lots. So, and this is Alon Simchovich, who is now an intern in the Sheba Medical Center. So that brings us to what we do these days. So we are looking at the impact of anxiety at different age times in mammals. So let me tell you about these mice. If adult mice want to humiliate a little one, they would pluck the whiskers in one side of their mouth. That's it. And those mice become depressed for the rest of their lives, and nobody knew why. So what we do know is that the whiskers are innervated by nerve cells in the brain at the other end, the other hemisphere. So we knew where things should happen. And we decided to check why are these mice humiliated, how come it takes so long, and is it related to the genes expressed in those cells, in cholinergic nerve cells? This is a cholinergic area. Or is it related to the structure of cells? You know, a nerve cell has a cell body, processes that could be long or short and diverse. So this is Nadav Yayon again, and what he developed is a method first to label the nerve cell, the cholinergic nerve cells, and then to find out their structure, develop an approach to take them out, find the sequences, and ask how come this happens. Okay, and the paper appeared last month. So these are the cholinergic nerve cells in the mouse cortex. And I know it looks as if this is a very dense network. It's not. There are very few cholinergic nerve cells in the cortex compared to other nerve cells. But the method that Nadav developed enables us to follow both their structure, extract them, and look at the sequences. So when we look at the sequences, we find specific genes that are expressed differently. And when we look at the structure, we find that these cells change their structure from having one pattern of many short and one long to one and one. So there are changes in the network that stay there for months, which means it will be there for the rest of one's life. Or in other words, be careful with the young ones. Never stress a young child, because that might have a long-term impact. And if we grow the cells in culture, we could see the difference between those cells with many or one dendritic extension. Okay, so what about anxiety links with inflammation? How can we find those regulatory genes that control that? And this question asks how backup systems can become emergency operation plans. So these are the small RNAs that we talked about. At the same time, each cell in our body has transfer RNAs. Transfer RNAs are those cloverleaf things. They bring amino acids to the developing protein chain. And when they're done, they're broken down by nucleases to bits and pieces. That was known ever since I was a student, except no one realized that they can do a job when they're broken. And this is what we're studying now. So not only microRNAs, which I was so excited about initially, but transfer RNA fragments. And of course we do that in men and women cells in comparison. Okay. And we do it in ischemic stroke patients. So men are more susceptible to stroke than women. And they die not because of the stroke, they often die because they develop pneumonia, meaning that a stroke patient becomes very vulnerable in terms of their immune system. There are changes in the blood, in immune proteins. And Kasia Winek is an MD PhD who came from Berlin to the lab. She was a postdoc, and she came to me with this RNA sequencing of blood cells from stroke patients. She said, doesn't mean anything. And I looked at it and I said, but these are fragments of transfer RNA. I was exposed to that as a PhD student, so I remembered the sequence. And we decided to look at those as a potential drugs. Then we find papers that some transfer RNA fragment is essential for sperm development. Another one is absolutely needed to produce ribosomes. And they are all related to changes in a nuclease, angiogenin, which we knew is elevated in stroke patients. So now let's think about that. I was so excited about microRNAs because they're small. You only need to transcribe them and send them and blah, blah, blah. But that takes 7 hours, we just saw that. Whereas if you have a nuclease that is high up, all you need to do is hammer on that transfer RNA tRNA and get the fragments, which is an immediate response. So this is a tool that nature had developed to be an emergency tool. And let's look at what happens in stroke patients. Those microRNAs, the red dots, that are affected in the cholinergic system go down. They give space, and transfer RNA fragments go up. So we call that changing of the guards, and we did that before the coronation. Okay. So if that is the case, then we may have a new opportunity here to look for control systems and for potential drugs. And the question is, could that be used to avoid anxiety and inflammation? And again, our gardens at the Hebrew University in the winter, now it would look much clearer skies. And if we look at PTSD patients, we also see changes in volumes of parts of their brains. So again, this is a no going back disease. People with PTSD, I'm afraid, would suffer from that for the rest of their lives. And this is where we live. We live in Kerem, and we keep seeing this beautiful church. And I need to tell you that the world is really needed to change the focus of treatment to older individuals, because this was the world's population in 1950 by the United Nations. Very few elderly people existed in 1950. Now look at what happens in today's population, and the view on medical studies must change. So there was a study, beautiful study, in 1957 by a man named George Williams, who said that there might be genes that are very good for you when you're young, but could be damaging at an older age. He called that antagonistic pleiotropy. And this is Gili Moshitsky, who's writing a PhD thesis about that today. And I also brought you a joke about this clinician who's looking at someone and said, how do you explain your longevity? Is it exercise, diet, environment? And the guy said, oh no, it's genetics. I believe it is. But when we look at life expectancy, look at the predictability of life expectancy. Now, I ask you to ignore Japan because they don't have the next generation. Life expectancy is calculated including babies that used to die very often in the previous century, but still they're susceptible to a lot of dangers. In Japan, there's very, very little next generation, and their life expectancy on average now is about 90. But if you look at the other countries, there is an increase. We live a little healthier life. Alzheimer's disease starts today, 20 years after the age of onset that was known 25 years ago. So we need to respect that. So we looked for those fragments of transfer RNA in the cerebrospinal fluid. And this is Ido Paldor, who's a clinician from Shaarei Tzedek working with us. And when we looked at those fragments, we found that their levels decline with age. So could it be that they protect us? In blood cells, they would be refurnished because blood cells keep dividing. But the brain is different. We live with the same nerve cells we were born with and will die with that. So there is no replacement. So we wanted to look at those in Alzheimer's disease. Not in Parkinson yet. That would be for my next trip to Australia. And there are no photographs of Parkinson. For years I was showing a photograph of someone from London, and then I realized that photography was invented 2 years after Parkinson died. Anyway, so what about Alzheimer's disease? Dana Schulman just completed her thesis in the lab, and she looked at transfer RNA fragments and microRNAs in the cholinergic nuclei of brains for men and women with Alzheimer's disease compared to healthy controls. And the red ones are females, the black ones are males, and you can see here that transfer RNA fragments are modified because there are lots of dots up there, and microRNAs are not in females with Alzheimer's disease. And that happens in mitochondria. Now, mitochondria are little organelles in our nerve cells, in any cell actually, that control energy metabolism. And we inherit mitochondria from our mothers. So this is a mother-to-daughter transferred phenomenon. The problem was that we knew women deteriorate cognitively faster than men when they develop Alzheimer's disease, but nobody knew why that happens. And now we find a molecule, an RNA molecule, that its depletion might be the cause. It's definitely different from what happens in men with Alzheimer's disease. So we looked in the associated data, and we found that the link was not to brain pathology, but to the cognitive tests that those women were doing. So we just published that. It appeared on May 9th. So I was taking a flight that night to come to Australia, and I landed in Perth, and there is a telephone from Israel Times. Apparently there is a journal like that. I didn't even know. And they were interviewing me. I'm working in Perth at the airport. And it created a lot of reaction. Dana is very happy about that. And it's all about those microRNAs and transfer RNA fragments in the Alzheimer brain. Now let's go back further. And we didn't publish this study yet, so don't talk to journalists, please. But I can share it with you. And the question is, what happens in fetal development? Would fetuses for females be more susceptible than males to stresses put upon their mothers before birth? So what we do is we take umbilical cord blood from the babies when they're born, and we follow the stresses on pregnant mothers beforehand. And we map them, and this is what we find. This is Shani Vaknin. She's expecting next month, by the way. So most Israeli PhD students give birth during their studies. And these are baby boys, these are baby girls, and I hope you can see the left wing means decline. And boys don't show much of a change, and girls do. And if you look at both of them together, it seems like ignorant, nothing. So baby girls are more sensitive to stresses put upon the pregnant mothers before birth. What we don't know yet is if this protects them better or exposes them to more anxious reactions. We're following the clinical features of those babies now. So we're waiting for that. Okay, and it's also mitochondrial. So this is what I came to talk to you about. I need to thank all the donors of samples, remarkable, and my collaborators, clinicians and students, and the foundations that supported this study. The comparison between men and women is our current passion, and I think it's really important. And I need to thank my grandchildren who remind me how important age is. And I'd like to end by citing Marie Curie, who said that now is the time to understand things, not to fear them, but to learn what they mean, which is just brilliant, I think. And thank you for listening. Yom Yerushalayim Sameach. And I'd be happy to answer questions, and if anyone is interested, I always answer my emails too. Yes?
Thank you very much. It's very interesting. Were there any studies done comparing the Israeli donors or volunteers and Australia, America?
Well, the brain donors are Americans, actually. There is a significant effort run in Chicago by Professor David Bennett, who happens to have family in Tel Aviv, so I know him personally. And he, 40 years ago, initiated a study following the monks and nuns living in Chicago. Because in America, it's very difficult to follow one's address except when they belong to an organization. So they are promised medical care and annual checkups, and they donate their brains after that. So this is why David Bennett is one of our authors on that paper.
And is there a difference between Jewish and non-Jewish?
I don't know. These are all monks.
No, I just wondered if I was
So I actually...
He's a Jewish monk.
Yeah. I went to New York just before COVID, and I was giving a lecture in the aged home for Sephardi Jews. And they said they want to know the risk of Alzheimer's disease in Sephardi Jews. Mm-hmm. So they approached the Alzheimer's Society in the United States, and they got no answer. I said, sure, because they didn't yet look at the Mexican and black communities in America. Why should they look for Sephardi Jews? You want to study Sephardi Jews? Come to Jerusalem. And they did. So they support 2 fellowships in my lab. And the idea is to compare what happens, but we don't know the answers yet. Yeah. Yes?
Just a question. Those that have genetic anxiety and those that develop anxiety, is there a difference? Have you noticed differences?
I assume there is difference, but I didn't study that yet. We studied the opposite. That is, could mutations provide resilience from anxiety? And actually we did find a change in the acetylcholinesterase gene that confers resilience from anxiety and perhaps even interest in stressful experiences. Yeah. So carriers of that mutation, for example, tend to volunteer to be medical personnel in the army on their service. So they seek excitement, and they are not anxious when they're exposed to it.
Like paramedics.
Yeah, but it makes sense. You know, if there is something that damages, there would be something that provides resilience. Yeah. Okay, thanks again.
Thank you.
Wonderful. Thank you so much, Professor Soreq. I really appreciate it. And thank you everyone for coming. I hope you enjoyed it. Please help yourself to refreshments. And we hope to see you in our upcoming events.